Denaturation of DNA refers to the disruption of its double helix structure, causing the separation of the complementary strands. This can occur due to high temperature, pH extremes, or chemical agents, rendering the DNA temporarily single-stranded and enabling processes like replication and transcription. Denaturation of DNA occurs when this double-stranded structure is disrupted, causing the two strands to separate and become single-stranded. Several factors can lead to denaturation, such as high temperature, extreme pH levels, and chemical agents. When DNA is exposed to high temperatures, the thermal energy disrupts the hydrogen bonds between the complementary base pairs, leading to strand separation. This is the principle behind techniques like polymerase chain reaction (PCR), which amplifies specific DNA sequences. By repeatedly denaturing and replicating them, extremely acidic or alkaline conditions can also break the hydrogen bonds and lead to DNA denaturation. pH extremes interfere with the stability of the DNA double helix. Certain chemicals, like urea and formamide, can disrupt the hydrogen bonds, causing DNA denaturation. These chemicals are often used in laboratory experiments to denature DNA for various purposes.